Hello everyone. Welcome to this pre-recorded session of Evolving Trends in Indian STEM webinar series organized by Sci-ROI India. Our first topic of this series is undergraduate research and mentorship in India. My name is Bhargav Sanketi. I am working with Sci-ROI India, and I'm excited to have a conversation with Professor G. Nagarjuna today on key issues relevant to making undergraduate research and mentorship in India more accessible and impactful. Professor Nagarjuna is a science education researcher in Homi Bhabha Center for Science Education, which is with TIFR in Mumbai, India. He designs collaborative learning environments for the construction of scientific knowledge and is an advocate of the freedom to read, write, and act, particularly in the digital space, Creative Commons, and distributed justice. He will tell us about his efforts in situating STEM education in participatory engagements in the form of a community of practice and share insights on how such endeavors can nurture scientific temperament of young learners. Thank you, Dr. Nagarjuna, for joining us. We will start this conversation with knowledge. So can you tell us what made you start Knowledge Lab and how it came to be?
There are a couple of issues that were in my mind when we started the lab. One, of course, is access to knowledge, and also as the institutions, particularly the educational and research institutes across the world, are turning to use computers as a means of education. So the question of access to knowledge becomes more important because the issues related to digital divide become very important, particularly in a country like India, where a large number of schools and colleges hardly do not have any kind of access to, infrastructure. So, except for some elite institutes, the rest of the students and people are left on their own, and the system doesn't understand the political aspects of this space. So keeping those things in mind, we started this lab with an explicit premise that software that we ask teachers and students to use must be free and open source software. It cannot be proprietary software because of the various kinds of things which actually go against the democratic norms of the society and the social justice. So that was one of the reasons why we named our lab. We replaced K with G in the spelling of the lab. Maybe I can just quickly share the website, which basically tells you why we are different. So let me show you the lab. I hope you can see the lab website.
I can see it properly now.
So this is so that's what I was mentioning. So this is it's called Knowledge, but spelled with G and OW. Essentially to reflect the philosophy of the GNU project, which is also being shown in the next window. So this is the free software project that our lab adheres to. And also, those of you who are interested, please do read the Knowledge Manifesto, which is also been linked on the opening page. It was a kind of a declaration of several friends. We got together and decided to put together this idea of the free software. Sorry, so I'm just missing out. Where is the Zoom link here? Yeah, I found it.
Yes.
Yeah, stop the sharing now. Yeah. Okay, so that's basically the aspect about the lab. And the lab started sometime in 2001.
Mm-hmm.
And so we have been active for more than a decade now.
So how did this team really come together? Like what what positions were you all in when you thought that this was something that you wanted to start up?
You see, so apart from the digital aspect, the science is supposed to be done, not read.
Yes.
That's been our main way of doing things. See, and also the other theme that is very important for us is that we want to produce producers, not consumers in the society.
Right. Mm-hmm.
Okay, so and these are some of the important themes that we took as axiomatic position of the lab. So just as we use free software for the same reason that you can be a co-producer. So while you're learning, while you're using software, you also start becoming a contributor to the free software, because it gives you access to the nuts and bolts of the technology that is given to you. So that has been the premise of our lab. So our lab uses this aspect that if you don't repair a device, if you can't repair a device, and if you don't have permission to repair the device, don't use it. So we use that repair philosophy. Right. And therefore, naturally, our lab is inclined to do-it-yourself kind of models. So we try to encourage people to make their own lab instruments. Whether it is a spectrophotometer, colorimeter, or incubator, or whether temperature measuring, whatever they want to do. For example, we encourage them to actually make it themselves, because this is one of our main context. Because we don't want to do it like physics, chemistry, biology, and some other subject. We want to do it saying that science is not about the content, science is about the culture.
Right.
So this is the third aspect of our lab's important assumption, that we focus on culture and not on the content.
Right.
Because the moment you get into content, you talk in terms of terms, their definitions, you remember some theories and models and things like that. But you don't actually understand that being a scientist actually is a lot to do with developing certain attitudes towards the world. Like, for example, one example could be seeking evidence.
Right.
Like you hear something, and then immediately you ask this question: do we have evidence for this? What is the support we have? Is it an empirical support? Is it a theoretical support? Or is it because somebody big man, somebody great has actually said it? Mm-hmm. So what is the basis of why we believe something? So these are the important aspects. And this is a lot to do with the culture.
Right.
It has nothing to do with whether you are a biologist or you're a physicist or a mathematician or something like that. Right. So that is one of the reasons why we developed a notion of what are called STEM habits. Again, as I said, since we don't want to distinguish on the basis of the subjects, mm-hmm. So what we're actually talking about is science, technology, engineering, mathematics have to be done together.
Right.
So that is why we looked at what would be the best model for integrating everything. So we thought of two important things. So one is investigation. Mm-hmm. And two is innovation, which essentially means making.
Right.
So either you're fabricating something, or you're making something, or you're investigating something. Mm-hmm. Of course, investigation also requires you to make certain things as well. So this is the way how we want to put it together in a form of a kind of a pedagogy. So a pedagogy that doesn't allow you to investigate and fabricate, make, or innovate is actually going to produce only a consumer and not a producer.
That's very true.
So what we're articulating is not necessarily only for Indian subcontinent, but it's a kind of a global concern everywhere. That science education is broken throughout the globe because everybody looked at content and not at the culture.
Right.
Because though India is the only country where the constitution says that every citizen should develop scientific temper, but which essentially means developing STEM habits.
Right.
So what does it mean for a student to develop, either you call it scientific attitude or scientific temper? So what does it mean? So our idea is that it actually means that you develop those STEM habits.
Right.
Like suppose when someone is speaking, okay, can you find out what are the assumptions that the person is making while he is speaking?
Right.
So try to identify what are those assumptions, or which part of his articulation is a speculation, which part of it is actually depends on some kind of a reasoning, some kind of a derived understanding. So these are the various aspects about thinking scientifically. So keeping that in mind, we launched a project called STEM Habits, or STEM Chat or something like that. So before I go into that, I wanted to touch upon another important aspect that we talked about, which is that as you see, the moment you get into a classroom, you have to listen to a lecture. And then when you start talking, the teachers normally discourage you to saying that stop talking, because this is not a place for you to talk.
Right.
So it is really ironical that if you stop conversation, you stop growing your mind completely. Because you can't encourage people to get into a conversation. That means it's not an academic environment, period. So that's the kind of philosophy that we follow. And therefore, we want to make sure that, how do you put it all together? And so considering that, we are already into a digital space. So what we did is we created a portal, a kind of a platform for people to get on to all those things.
Right.
So maybe at this time, I'll just quickly show you that portal.
Yeah, that would be great.
That might be a better way of inviting people to our way of doing STEM education. So let me see how this time the sharing screen actually works. All right. So hope. So metastudio.org is the name of the site.
Mhm.
Can you see the site?
Yes, I can.
Yeah. Okay. Now, as you can see, it actually says that welcome to Chatshala, not Patshala.
Yeah.
Yeah. I know that's the pun with the language out here. So basically, to say that we are here to chat.
Yes.
And we are here to take it with a lot of fun, and it's informal. And so we also want to call them as STEM games, because there are some rules that you follow.
Right.
And then what we do, of course, is that there are two main activities that we do. One is the makerspace activities, and the other one is a cube project, which I'll talk a little bit a little later. But what I wanted to show here on this platform is that the platform actually includes a lot of chat. Like, for example, there is something called STEM chat here. Let me make it a little smaller.
So who is the audience here, Professor?
So the audience, anyone who is about 13 years of age.
Okay, I see.
Is an audience here. So you have different kinds of chats open for different subjects, like the makerspace chat and software freedom chat and things like that.
Right.
And there are blogs and wikis and things like that also that people can do. And there are regular questions, like, for example, there is a post here about mosquito mapping. There's a post here about studying microbes found in human body. Why would dog attack a puppy? For example, we are studying the behavior of mammals around our environment, things like that. So these are the things that are posted by students, mostly undergraduate students. But we do also have a good number of school students as well. So these are this is the kind of discussion space that has been created. But most important thing that I wanted to talk about is the badges, because that is where we're talking about where we are focusing on culture and not on content.
Right.
We never give anything any mark or any credit when people speak things correctly. But what is the kind of culture that they're developing?
Right.
So people are granted these badges.
So they take initiatives, they get badges, right? Like, so
right. Like, for example, I'll just give you example of STEM habits. So these are grouped STEM habits. So there are about 70 to 80 STEM habits that we talked about.
Right.
So if you look at the first one, for example, when keen observers are, they see more than what normal people can see in a phenomenon. So as soon as when people report that and they talk about it and they write about it as a small microblog, they may be given this badge.
I
of a
see.
key node. Okay, and suppose when someone declares assumptions and defines the terms and states a set of postulates to build a model or a theory, he gets a badge called axiomatic.
Nice.
But as you see, nobody so far got this badge as of now.
Maybe it's time for me to get on this website and get this badge. But
See, so
that's
yeah, you should get onto the site and maybe give us an opportunity to see, in what kind of a conversation are people able to find out this, because axiomatic thing is the beginning of science, right?
right.
If you
Yeah.
look at Euclid as an example, like if you take Archimedes as an example, these are the people with when we started science as a whole, right?
Yeah.
As a beginning, and they started with axiomatic understanding,
Yeah.
and now we don't even talk about it when we are telling people about what is your assumption, what is your definition, what are your postulates, and how is that reasoning actually working. So this is what I mean by focusing on habits and not on the
Right.
content.
I
And for example,
completely agree.
yeah,
Yeah.
similarly, I mean, I'm just going through a few of them. Like there are times when suppose somebody is collaborating with other person,
Right.
want to join in a project, though he gets a collaborator badge.
I see.
Sometimes somebody gets a critical thinker badge who looks at a problem from multiple perspectives rather than and asks questions and assumptions. And somebody is a data collector, somebody is a designer, doodler, for example. Somebody actually makes a small sketch to represent something, and somebody is diligent, so because he's very meticulous and he takes a lot of care while performing his work and
Right.
things like that. So this is the way how we said, like for example, somebody is a modular thinker or things like that. So people can go through these STEM habits on the
Right.
site, and I would consider this as one of the really cool. Uh, I would say a kind of a model which is we consider as an alternative to anything that is happening anywhere in the world when it comes to STEM education.
Absolutely.
The reason
Absolutely.
being, the reason being that everybody is focusing on content, but not on these skills,
Right.
uh,
Yes.
particularly the habits that we're talking about. So this is the the kind of, so we are what we're doing, why we are doing. There is some philosophy behind it. We may be completely wrong, but the time will tell whether we are right or wrong. But this is a kind of an experiment that we are doing in the lab, and so one of the uh, cool projects that started and inspired us to do all these things
Mhm.
is called the Cube project.
Right.
So the Cube is called the Collaboratively Understanding Biology Education.
I see.
It's an expansion, it's an acronym
Yes.
for Collaboratively Understanding Biology Education. When we started with undergraduate students.
Mhm.
So that's how we called it initially, as collaborative undergraduate biology education.
Mhm.
But eventually, when a large number of school students actually joined,
Right.
and then we figured out that undergraduate students learn better when they're in the company of school students,
Interesting.
and school students learn much better when they're in the company of undergraduate students. So we completely broke the traditional model of, you know, keeping in a classroom all the idiots of the same age. Sorry to use the word idiot, because I find it's very idiotic to. I mean, I'm criticizing the pedagogical model that we have adopted.
Right.
It is idiotic to have all people of the same cognitive level, so to speak, in the
Right.
same classroom, because there is no potential difference.
Mhm.
So
Yeah.
who will ask the question,
Right.
or who
Yeah.
will be the person who will be there to guide and mentor the other people who, if they are doing anything. So that is why we adopted in the Cube model a kind of heterogeneous kind of thing, where we keep school students, undergraduate students, and postgraduate students in the same group.
Right.
So
Yeah.
that's been our experiment again. So this is the way how we are experimenting in a very non-traditional way. So in the Q project, because it's biology, right? So we need a context. See, for other science subjects like physics and chemistry and other things, like we use the context of measuring something, or understanding a phenomena and investigating something and all that. Of course, in biology also we have to do that, but biology interestingly has a very interesting thing called model systems. Like earthworm as a model system, fruit fly as a model system. I don't know what your subject background is, but
I use chicken and mouse models to study developmental biology.
Wonderful. So you speak
I
our
totally
language.
get that.
So where do we find those models? Like for example, suddenly somebody says, you have to get Drosophila melanogaster, but I get fruit fly in my kitchen.
Yeah.
Is that actually Drosophila melanogaster or is it some other species?
Right.
Did anybody note it? So who is there to find out? So we give that as a small activity. So why don't you go and capture them? So what actually happened, of course, is that the cube started in 2011, and we are almost like close to 9 years now. And these activities have been happening, and we have thousands of students across the country participating. And during the pandemic, they had no labs.
Right.
And they have no access to the labs because the colleges are closed.
Yeah.
So what do you do? So our cube, funnily, had flourished much better than when the colleges were open.
Right.
The reason being that since our model is so open and so mundane and so down to earth, they're able to find model systems in their regular household.
Yeah.
All that they need is a fly, or they need a yeast, they need a bacteria, they need a fungi. All of them are in plenty around in our household.
Yeah.
And they started investigating them. And they didn't have the lab, they didn't have the chemicals. Then they found the chemicals in their own house. And they started working with it. And some interesting innovation started happening. And I think I must show you this in important investigation that happened. I'll just show you a couple of pictures, and through these pictures I will introduce you to the cube model.
Yep.
So this is just a Telegram group that we use for a lot of things. So let me take the cube. So we have this MetaCube group where we have archived some interesting pictures out here. So let me show you this one. Can you see this picture?
Yes, I can see it.
Can you see this picture?
Yes, I can see it.
Okay, so as you can see, these are regular mineral water bottles. And they're not Borosil bottles that we use in the lab.
Right, yeah.
These are inverted bottles. And you see there is a medium on the top. Actually, then the bottom of the bottle there is a medium. And what is that medium? Normally in a regular biology lab, you would use an agar-agar to solidify, to create the medium. And you'd use propionic acid and fructose and glucose and other mediums, and you actually prepare medium in such a way so that the fruit fly can be grown.
Right.
But this one doesn't look like agar-agar. The reason being that this one actually, this of course is the same bottles that have been put to here. So this is the innovation that happened during the pandemic by our cube students. So they have used a tomato, okay, and rava. You know what rava is?
Yes.
Possibly.
Yes.
Yeah, which is what we use for making idli and upma.
Yeah.
Okay, so that rava is used as a food replacement. And then we used vinegar to maintain the pH as well as also to preserve so that the other infections don't happen inside the bottle.
Right.
Okay, so this is what we called it and named it as a TSRV medium. Of course, because we added some sugar also in it.
Right.
So tomato, sugar, rava, and vinegar.
TSRV.
So this is called the TSRV medium. And using this, we have sort of developed the whole thing. And then this is another model system that we use. This is a water flea. You go to a pond, collect the water flea and start breeding it. So that's another model system that is being used. So this is the these bottles actually have those things. And then this is a plant model system, which is just a something that you will see in the ground anywhere when you walk out.
Yep.
And they're studying the behavior of this plant. For example, day and night cycles. The diurnal cycle is very clearly visible in this plant. Bhu amla, as it's basically the same species as the amla.
Right. Yeah. No, that's what I thought. The leaves kind of look similar.
Yeah, but this is a herb, but not a tree.
Yes.
Okay, so this is called Philanthus. The Philanthus is the genera. Okay, so the interesting thing is that if you actually touch the back end, a little bit and do a little bit of irritation, the leaves actually close.
Oh, I see.
As if it's like touch me not.
Touch me not plants. Yeah.
But touch me not plant is very sensitive, whereas this is not very sensitive. Okay, but then it shows the similar behavior. So now people are studying that, that kind of a behavior and trying to see what is the time of latency for the wake up and sleep cycles and things like that using this kind of model. And and this is another model system. You may be knowing the very famous model system in plant, very widely used in molecular biology. But whereas this one is its relative.
I see.
Okay, so so this is very widely again used, available as a regular. Just it will grow in any. If you start walking in any of the gardens, you will see it as mostly as a weed. And this weed is now been collected and put together by the students, they're growing them in their own gardens, and actually study the floral bud. They use the floral bud and actually do biotechnology experiments using this by dipping the floral bud in the in a solution of DNA, and actually try to introduce the DNA from the the bacterial genome into the plant genome.
Wow.
So these are the kind of experiments that the students are actually doing and investigating with all that. Of course, this is another way of extracting things from the. This is a fruit fly in its life cycle. And the other thing that we also tell people is that that you are not just doing experiment, but you have to label every sample.
Yes.
When do you start? When did you prepare the medium? What is the label that you do? So this is another habit that we call as recording habit. See, you can't do science without recording.
Absolutely. Yes.
And so this is another thing, like we teach them, how to develop these things, and you can't develop them. You can't develop this kind of habits by reading a book, right? You
Absolutely.
have to do it. So that is what we meant by, developing STEM habits by actually doing, but not by reading them in a textbook. So as you can see, these are so meticulously kept here. And the TSRV represents the kind of medium that they're using.
Yeah.
So they put the date and they have the thing because they develop single line cultures. So, which essentially means, they just take one gravid female and let it lay eggs and then collect all the offsprings of that and then transfer them to other bottles and things like that. All this, let me tell you, is happening at home.
Right. Yeah. No, that's amazing.
These are home labs.
Yeah.
These are home labs. Okay. So these are home labs. And these are the various model systems, of course, that we are using. I was just talking to you about the Philanthus model. So these are the pictures that are done by them. The students do these things using their mobile camera. Okay. They're not using anything because they don't have a microscope. They don't have any sophisticated instruments.
Right.
So they're all using mobile cameras to even do a cross section of the stem and try to see as much as possible all the details with just using the mobile camera itself.
Yep.
And look at this. For example, we talk about the cube Chatshala happens every evening. In fact, it is happening. It must have closed at 9 o'clock today. So it starts at 5:30 in the evening, and 9:30 in the night. So it's about 4 hours. There's a continuous chatshala where students who are working in the home labs come and just chat with each other.
Wow.
And while they are chatting, they draw this collaborative plot. So as they're speaking, their observations are plotted, and they create these observations, and they develop this STEM habit called graphicacy.
Yeah.
Because when you collect a lot of data, you have to represent that in a graph.
Absolutely.
You have to organize the data in order to understand what is the pattern, if there is anything in the phenomena. So how do you observe phenomenal patterns? How do you model them? So this is model building and model-based reasoning and things like that, which sort of develops. So this again is not something that it will be happening by just looking at one good plot here and there. So these are the kind of labs, examples of lab activities. So this is a very crude example. As you can see, there is a potato cut at the base here. And you see some soil kept around the potato rim.
Mm-hmm.
Now, what do you expect from this small experiment? So every soil has nematodes.
Right.
Okay, so the expectation is that, at the center of this potato, you put a small drop of sugar or something like that. The nematodes will concentrate and come to the center. And then they collect those nematodes and then start doing these experiments with them.
Wow.
So which nematode is that? Is it C. elegans, or is it a cousin of C. elegans? Right. Or which is that nematodes that are there in the soil nearby? So extracting such things sort of happens. And this is another whiteboard work that happens in a chatshala where they're discussing about the nerve reflex and what is the chemistry, how are the ions and polarization of the membrane actually produced and things like that. So this is so what we're doing essentially is that we are providing a context.
Yeah.
So that the biology content can be introduced.
Yeah. No,
the content doesn't come without a context. So that is our philosophy of education. There's always a context in which the jargon is imposed. So you don't start with jargon, but we start with a context, a very simple context. And then we investigate, and then people get on to those things. Of course, I can go on like this without, there are plenty of pictures, that talk about it. So this is about discussing about the ganglions and the expanded wings of the Drosophila. And this is about earthworm regeneration experiments that are being conducted about, for example, so they're actually studying, they actually study the regeneration of the ventral nerve cord through these experiments. So this is another place where they're actually extracting the DNA at home. Yeah. And so this is another experiment where they're extracting DNA from a banana pulp. So this is the same Philanthus amarus, debilis, and ulinaria. As you can see, they are not only collecting these different kinds of Philanthus, but they're actually able to do the taxonomy. Can you see that?
Yeah. Yeah. Absolutely.
So they're actually seeing that they're actually able to find out that there are different species of the same genera.
Yeah.
In the environment, and they're able to do that. So this is doing proper biology, and actually most of our students actually think that they're scientists.
Yes,
because they're actually doing this,
which is very important.
About nail regeneration activity that they're doing. See, I can go on like this. There are a lot of this.
But I definitely get the idea, right?
But everything is from your neighborhood area. As you can see, this is just some side of the road. There is some drain, and you collect some herb from there and algae from there or fungus from there, and you start working with what you have in the kitchen. Potato could be your medium, right?
Yeah.
You don't have agar-agar plates. Well, potato is available to you. Yep. So a potato is an agar-agar plate for you in the kitchen, and you start using that to do these experiments. And as you can see, this is exactly how we are promoting. Basically what we wanted to say is that, if you want to become a scientist, and if the labs are not accessible to you, don't worry.
Yes.
Sophistication is required in the mind, not in the lab.
Absolutely.
So this is the kind of philosophy that we've been talking about.
So I have another question for you, Dr. Nagarjuna. I think you answered most of my questions through the course, but I had one last question for you. And I really like the idea that, science is the process, not necessarily the information, right? So it's important to learn the process when learning science. So a lot of the members of Sci-ROI, people who interact with Sci-ROI India and Sci-ROI Global are actively engaged, with teaching undergraduates either in India or outside, and also research and mentorship and things like that. So most of your programs seem like supplementary programs, right? So they have to keep going to school or keep going to colleges, but this is like supplementing that education with actual science process education. Do you have any ideas as to how members who are will go on to become professors or will go on to mentor students, how they can take some aspects of this and inculcate this into their own teaching pedagogy? Like, do you have any suggestions or
one of the most important things is actually that we can't change the education system overnight.
Right.
In fact, if there is one social system which is very robust to change, and that is called academics.
Yeah.
They're very, as if, they have already decided what to do.
Right.
They don't want to change. They have to follow the syllabus, they have to follow the curriculum, they have to follow whatever not. And it's a mindless model that they've developed without understanding that education actually means change of behavior. If there is no change, and if you can't accept that academics actually support change. So what is it that we are doing? Like, as I said, the cube is being done from 2011.
Right.
Okay, now that means that we were working in 2011 with 12 undergraduate students, of which 4 of them are teaching in a undergraduate college.
Wow.
As teachers.
Right.
And now they're still working with us as mentors, though they are teaching somewhere else. And so, we are very patient and slowly trying to restructure and inform people about, why we are so immune to change. And why the hell are we saying that, I have syllabus to complete? And come on, the only syllabus that you have to complete is give them the STEM habits.
Yes.
Make them scientists, make them producers, make them creative investigators.
Yeah.
And let them see that there is science in everything around you. And you should be able to wear that, the goggles of science and things like that, and start looking at the world with a different paradigm shift. So how do you produce that paradigm shift in the way how they think about it? So that's why we are going slow. So the answer to your question is that unless there are programs of this kind, and unless teachers and students are exposed to them, it doesn't happen. So I'll tell you why we are focusing on undergraduate students. The reason is very simple. The reason is that today's undergraduate students will be tomorrow's teachers and professors.
Absolutely.
And I can tell you one thing. All those thousands of students who are now part of the cube, even if out of those 1,000, even if 20 of them become college teachers tomorrow, or even school teachers tomorrow,
that changes everything.
I can promise you that they will not go to the classroom saying that I have a syllabus to complete.
Absolutely.
And that is the change that we want to bring in.
Right.
We're patient, we're slow. We just want to focus, because culture change is not easy.
Right.
Remembering some content and regurgitating content is easy in an exam, but transforming yourself and restructuring your thoughts and thinking differently is not easy. And that requires support from the system, support from the family, support from everywhere. Let me tell you one thing. There's some important change that is happening because students are actually doing investigations at home. Mm-hmm. The mothers and students and brothers and sisters are asking them the questions.
Right.
And the students are actually asking them those things.
So it changes things from the grassroots level.
Yeah, there is one small story I wanted to tell you. Is that one of the students from a small slum area here started collecting mosquitoes at home. And the objective being to find out whether it's an Anopheles mosquito or is it an Aedes mosquito, because they need to know whether the mosquito will give you malaria or it will give you dengue.
Dengue, yeah.
Right. So now there is very easy to identify the dengue mosquito because it has got white scars on the legs. So it's very easy to find out because like a tiger mark, like on the skin that you have that. So this was the activity that the child who is an 8th standard student doing at home. And when the student went to the school, after she returned back from the school, this incident happened about 2-3 years ago. And the mother in the home gave her a packet of a newspaper thing. And when the daughter opened that packet, she found a lot of mosquitoes. The mother was collecting mosquitoes for the child. And then the mother also can know the difference between Aedes and the Anopheles. Now,
Right.
this is the advantage of not closing the walls of an academy.
Yes.
So school investigation, science and mathematics, if it happens only in the school and not at home, there is a danger.
Yeah.
Because the horizontal transmission of knowledge doesn't happen.
Yeah.
So there are a lot of advantages if we actually say that our academies are living academy.
Yeah.
So that has been the way how we are looking at this whole model. Of course, we're not in a hurry. We're happy to, but the interesting thing, of course, is that because we do it as a game, a lot of people actually come, they get motivated. And the transformation that you see in them, you can see the glittering eyes that they have. They become very good conversationists over a period of time because we encourage them to converse. We never say, shut up, this is not the time for you to talk. Always talk first and, write later. And, that's the way how we make them. And all of them become very good speakers. And they talk and they also write well. And over a period of time, they're writing good blogs as well. And
Right.
they know what to write, how to write.
And
when
that
they
is
share
the
a
scientific
picture,
process, right? That is the scientific
absolutely.
process.
When they share a picture, like you see, in every WhatsApp and every social thing, they keep sharing pictures. And when they share a picture, they don't tell you the date, time, place, nothing. They just share the picture. But our students don't share the pictures like that. They tell you what is the latitude and longitude where the picture was taken.
Right.
Okay. What was the time when the picture was taken? And what do you see in the picture? And what do they see in the picture? Maybe someone else will see something else there.
Yeah.
And so that becomes a context for a conversation.
Right. That is amazing. And I'm definitely amazed by all this. And I think that's definitely the way forward. And I think, people who are going to be listening to you either in this talk or during our webinar will take away that, the inculcating STEM habits is definitely very, very important, and bringing that to the school level and to the undergraduate level, and not just focusing on the content, right, is going to be the big change in ideology that would help us become better at research mentorship. And
I'll be very happy if that is what the listeners will take back with them.
I hope they do. Well, that concludes our conversation for today. Thank you so much, Dr. Nagarjuna, for your insights on this. And we are really excited to continue this discussion during our live panel session on August 26th, 2021. And,
Sure.
Everybody can ask you their questions at that point of time. Till then, goodbye.
Yeah, goodbye.